way of thinking are the ones that will best allow us to choose and parameterize a model. So what you really want to be able to do is, for an experimentalist who's just discovered some new phosphorylation state of an ion channel or a new mutation or something, you want to be able to give them the experiments that they're going to need to do to capture the behavior of that ion channel. And then you're going to want them to see the data that comes with as a result of doing those experiments. And then you don't really want that to just float there. You want to know exactly what you've got to do to replicate the best model for that ion channel using this new data set. So the new data comes in, attached to the protocols that we use. How do you actually come up with a model from the data? And if you can automate all that, there's no reason this cycle of experiments and model building can't be done in days rather than months and years. But we'll talk about this more at the end, and you can shoot down my ideas. And then, when the virtual physiological human's in use in every hospital in the world, you can just upgrade the virtual physiological human. It can start making better predictions for people with this mutation in the ion channel. And it all should be almost an automated process. So what's actually missing from this? We can email experimental data across the world instantly. So that's not a problem. The experiments at the moment probably take a month or something, but that's probably arguably not the rate limiting factor in all this. We don't really have a virtual physiological human, but perhaps if we made models like this, we might do. So I think it's this idea of sharing the experiments that you want to be able to do, attaching the data to them, using the experiments and the data to make a model, and formalizing how we do it. So we've tried to kind of chip off a little bit of that and just think about how we share what experiments we want to do. So you're probably all aware of brilliant efforts that Peter and the gang have done with things like the Physio repository, which stores CellML files, and there's also the SPML files in the biomodels database. So these are brilliant. There's thousands of models that we can now share. There's no ambiguity about what the equations are. But what these tend to look like at the moment, if this was a model repository, there's lots of models in it. But each model really contains one single hard-coded protocol. So if you load it up and press go, one thing will happen. Typically with the cardiac models, that would be 1 Hertz steady pacing if you load up the CellML file. So that's much more useful than not having a model repository. So we think that it's a brilliant thing. But the idea of all this web lab is to separate out that model and protocol. So this is the sort of picture of things. So we separate out the model, which we still have as a CellML file in this system, from the protocol that we want to apply to the model. So in a simple case, this just means there's a 1 Hertz pacing protocol, a 2 Hertz pacing protocol. But we can do much more complicated things that Jonathan will show you, like get IV curves for ion channels, do restitution curves, that kind of thing. Then the simulator becomes something that picks a model and a protocol. You need a language so that these understand the different components. So a protocol might say this is going to be the stimulus current. The model will have to say this is where the stimulus current would be applied. And so that's it really. It's not a very complicated concept. Does that make sense? It'll make sense when we have a look at what it all does. So key features of the system is that once we've got this protocol, that's independent of the model. We can apply the protocol to any different CellML model. And the interface is described at the level of things like voltage, current conductance. A nice thing, thanks to John, is that units conversions are all handled automatically. So if you've got one model in picoamps, picofarad and another in just amps, we'll try and use the capacitance and everything to do the units conversions. So you can actually compare, nearly all of the models we've come across can be compared directly. Another neat little feature is that the protocol will specify the inputs and outputs that it wants to apply to the model. So we don't need to solve all the equations if they're not needed. For example, if it's an IV curve, we could say the input is voltage, the output is the sodium current. The system will work out that it doesn't need to solve most of the equations. You can just figure out which one wants the sodium current, and it'll work with those. So you can replace bits of models if that's what the protocol does. For example, just encoding your own stimulus current instead of the one that's in the model. In the protocol description, we've put all the post-processing and plotting that you need to do to reproduce a graph that you might put in a paper after applying this protocol. So an IV curve, an actual potential trace, that kind of thing. And there is the ability to do some fairly complicated parameter sweeps to construct an IV curve clamping to different voltages, that kind of idea. So I think that's my motivation. I'll hand over to John to tell you some of the details of the protocols.
Just going to give a very high-level overview of what you put in a protocol description for now. And this afternoon, you'll get to dive into this in a lot more detail and start developing your own protocols to look at different kinds of experiment. But the main things we have in a protocol sort of follow the natural flow of what you would want to do in an experiment. You're starting off with inputs for the protocol, which can allow it to be specified for different situations. So you can have a generic steady pacing protocol, and an input to that is what pacing rate do you want to apply to the system. You've then got the simulations of the model that you're running, so the raw time courses, but also nested loops if you want to run multiple different time courses, starting from a particular condition, or arbitrary nesting and structuring of different simulations you can put in. Post-processing of the raw simulation outputs to actually compute what you're interested in. So generally, it's not the raw time courses that are most interesting, but it's some quantity derived from those, such as the actual potential duration. And then what the outputs of the protocol are. So what of all the quantities that you computed in the raw simulation data is of most interest in this experiment. And some of those outputs you can then decide to plot in different ways. Some of the key bits here, possibly the most important bit in the system, is this interface definition component, which says how you plug in any model of a cardiac cell into this box in the middle of your simulation loops. So as Gary was saying, specifying what the inputs of the model are that you want to be able to change from your experiment, what you want to read out as outputs from the model, and how you're doing unit conversion, any alterations of currents, and that kind of thing all come within this interface definition part. And one of the nice features about the system sort of abstracting is that your model doesn't necessarily have to be a cell-ML file representing a cardiac cell, although a lot of the interface stuff works on that assumption. But actually, you can plug in anything that has inputs and outputs. So you can plug in as a model an entire protocol, an experiment run. And that allows you to do much more complicated setups with nesting post-processing inside simulation loops. And we'll see quite a few examples of that for doing things like restitution curves and drug block protocols. The nested simulation loops is the other key bit, enabling you to do multiple simulations or sequence simulations of your model and getting out as your outputs, the sort of core data type throughout the whole system, n-dimensional arrays of values. If you do a time course simulation of a model, you just get a vector, one of the vector of values for a particular model output. If you then nest that within a loop, you get a 2D matrix of values coming out. If that's nested within a further loop, you get a 3D array, and so on. So the basic data type that all the operations work with, and especially all the post-processing works with, is this n-dimensional array structure of values coming from your models. And then plots, the other key thing, specifying what you're actually going to display as the human consumable output from the protocol, and what is typically going to be compared between different models for different protocols as well. And there are various other bits, and we'll talk about more libraries for defining functions for use in post-processing, the ability to import protocols with functionality that you want to reuse, so that you don't have to start from scratch every time, but can build up protocols quite quickly based on what's already there. And a little bit more of an introduction to that interfacing topic, where you've got your models, your protocols. You want to be able to apply one protocol to any of the models in the system. And you immediately come up against a problem in that different modelers have decided to use different variable names representing the same quantity. And voltage is one of the most variable, with 5 different names in common use across the cardiac field. But most other concepts, you have similar naming variations. And so we use this idea of ontology to apply consistent naming across the system. This is basically a way of giving unique labels to concepts. So you can say, here's my label for the transmembrane potential that I'm going to use. And you can also, if you want to, describe the relationships between these concepts and sort of build up your knowledge of a domain encoded explicitly in an ontology. For the most part, we just use it for the unique labels aspect here. And the modeling languages like SLML have the ability to annotate variables with these labels. So we can annotate a variable in the model to say this is the membrane voltage using a term here from our ontology that looks rather like a URL. So it's got a base part to say that it's our ontology, and then a particular term within that membrane voltage here. So we can say in this model, V is the membrane voltage that we're interested in. And the protocols then can refer to these labels such that we can prepare any model for use with the web lab just by annotating the concepts that the protocols need to access. You don't necessarily need to have a blessed ontology to use our one, but you do need the models and the protocols to agree on what ontology they're using if that interfacing is going to work. So for convenience, we've developed our own for use in the web lab at the moment. Obviously, you can work towards using community standards for these kinds of things as they develop. And now, I'm going to show you it working. Hopefully, it worked an hour ago. Hopefully, it's still working. Of what the system actually looks like and what you can do with it. So here's the home page for the web lab, which tells you a bit about it, some quick start links to various places, and that image that Gary showed of what we're trying to do with it. There's a link up in the top right here for registering to create yourself an account, and I'd ask you to do that fairly early on in the practicals and let me know what your username is, so I can make sure that I give you permissions to run simulations on our servers and give you a dedicated process to do that, so you're not all competing for the same little bit of compute resource. But the main bit you'll want to look at is this experiments link up at the top, which shows you all of the public experiments in the system at the moment. And we have this matrix view where along the top, okay, don't use the pointer, rather, along the top, we've got all the different protocols that are available, down the sides, all the different models, and each cell in this matrix gives you one experiment result. So that protocol applied to that model. The color coding is basically telling you whether it was able to run. So green means that it runs and produces all the results that you're expecting to see. It doesn't tell you anything about whether this is a good model or a sensible experiment to apply to the model, but it says it can be done, and you can have a look at the results to see how good it is. The grey color says that actually you can't run this experiment, that the protocol is interested in some biology that the model doesn't express, or at least hasn't been annotated. And so you just can't apply the protocol to that model. Orange means it ran and produced some outputs, but not everything that we were expecting. Red means it just didn't run. Typically, it's failed to simulate for some reason because it just couldn't solve the ODE system. So we can click on one of these cells to have a look at some results. So let's pick Caro model and look at sodium channel drug block. And we get, when we look through to that, the default plot, the first plot listed in the protocol as the main thing you would want to look at. And in this case, it's plotting action potential duration, 90%, against degree of drug block, again as a percentage. So as drug block increases, you see first a slight increase in the action potential duration, and then a sudden drop off at 100% block. And you can actually have a look and see all the other files that were produced from that experiment and stored on the system. And a lot of these are plottable result data. There's other result data, other files as well, which you can explore. But we can look and see, for instance, what's causing the sudden drop-off in APD at 100% block. If we plot the final pace voltage trace with this graph icon here, that will then produce that plot from the saved results. And we can see that for most of the drug blocks, we're getting a normal action potential. When you get to 100% block down here, it's just not actually depolarizing the cell fully. So you get something that looks enough like an action potential to compute an APD, but isn't really. You can see what's going on and explore what's giving you the gross results that you see. From an experiment page, you've also got links to view the particular model and protocol that were run. So you can go and see the model description, look at the CellML file, explore what's going on. You can also look at the protocol, in this case the sodium drug block, and each protocol description has a bit of documentation explaining what it's doing, and you can go and explore the protocol descriptions that make that up in detail. The main one is highlighted, and then others are imported and used, providing cell underlying functionality. We'll go into that in a lot more detail later on. So that's showing the results from one experiment. One of the most powerful features of the system is that it lets you pair experiments. So if we take just steady state 1 Hz pacing, simplest you can do, we've got here a compare models, where we can select particular models we want to compare, or a shortcut to just compare all the models on which this protocol has been run, and you see the standard result graph for that protocol, but all the models now are overlaid. So each one of these traces is one model's behavior under this protocol. And you can see that for steady state long time courses, some models don't get into a nice regime typically because they're not conserving charge, and others are giving you a whole range of different action potentials depending on the species, cell type, particular model. Another way of comparing that is quite useful is this comparison mode, particularly if you're not wanting to display just all models for a particular experiment. You can easily select from the matrix which ones you want to compare. So you can sort of select arbitrary experiments just by clicking, but you can also select whole sets of models. So let's select a few human models, I think Randy, and we can produce a submatrix just showing the blocks for those particular models. So now we've, rather than showing the entire matrix, we just got a few of them. And again, we can pick out, say we just wanted the Caro models, and we just wanted to compare 1 Hz and 2 Hz pacing. You can easily pick out those. We've got now just 4 experiments that we can compare directly. And you can see for these 2 variants, the Caro model, what it's doing in 1 Hz and 2 Hz steady pacing.
And that was just integrated when you press the button, or you're calling a library?
So all the results are stored when you upload model and protocol. You can choose to run experiments. So you'll run all the available protocols on your new model. The results are stored in the system. So it's not actually running simulations when I bring it up. It's just loading the stored results and plotting. Yeah, some of the experiments take quite a while to run, particularly if you're doing sort of 11 different percentages of drug block, running 1,000 paces of the cell each time to see what's going on. Most of them are reasonably quick. So that gives you a little taster of what's available, and we'll be showing you more of the features as we go on. But I think before I show you too much more and you start forgetting, it'd be a good time for you to try it out and explore what you can find in the system. I just got a couple of slides before we get to that point, I think, to say that obviously at the moment, we've got models and protocols, we've run simulated experiments. The sort of obvious thing that's missing from the system is, do these actually correspond in any sensible way to reality? So sort of the big missing piece that we're hoping to add to the system is the link to experimental data. And really, we should have the protocols that we've got in here should correspond directly to experiments that you might perform in a wet lab. And the outputs that are generated from these protocols should correspond in a directly comparable way to data that you might collect from real cells. And you would then be able to put in comparisons here and see which models are actually reproducing this experiment that I've done. And we'll talk more about that and other plans for this tomorrow afternoon. Just want to mention some of the people that have been involved in getting this off the ground. Myself and Gary have been the main drivers behind it. We've also had quite a lot of work from Martin Scharm, Dagmar Wolfmann's group in Rostock. We developed the initial version of the website for us. Aidan, and in the front here, my default student who's working on parameter fitting, starting to bring in experimental data. Eric is a summer student who did a bit of the programming for the behind-the-scenes stuff as well. And various people have contributed ideas and inspiration. Some of them are sitting in front of me. So over to you. On your programs and also on the website for the workshop, you've got a link to a Google document with suggested tasks to have a look at to help you explore through the web lab. You've got descriptions, some background reference material if you want to have a read, and then different things that you can try out. But do explore, have a look through the system, see what you can discover, and what you might want it to do as well. There's a note here that we have an online feedback document which you can all edit. So do, as ideas occur to you of things that you would like to see the system do, add them to that document. Or if anything's broken, hopefully the students did a run through this last week and we fixed anything that's broken. If there are, do let us know. Do ask us questions. We'll be wandering around. Any questions now before we head upstairs and have a try? Yes.
Have you thought about the future enough to know when you won't have enough space to cope with all the models, all the parameter fittings, all the complaints about models not working here, models not working there? Is there forward thinking about that kind of question? It really requires a team almost, doesn't it, to maintain this?
Yeah, so we're looking to put in a grant proposal for the next stage of development, which would fund you to work in a team to support it. At the moment, it's just running on a small cluster of servers in the basement here, but the framework is developed in such a way that it could be run on Amazon Cloud resources or whatever. So we do have the ability to scale up.
Yes.
We have the funds to do so.
So this is a kind of bootstrap to get there.
Indeed.
Yes.
Do you have a facility at the moment where you've done a comparison, now you want to tweak a parameter on the models? You want to see what effect that has in comparison models. Can you then upload that result simulation to your database and have that?
Yeah, so one thing I didn't show in the demo, but each model that's in the system, there it is, you can have multiple versions of the model. So there's a C version list here. You can have multiple versions of the model, so you can upload a new version with a different parameter, rerun all the experiments, and then compare what they're doing between two different versions of the model. And there's also some tools in there that allow you to compare exactly what's changed between different versions of the model as well.
Do you foresee the parameter estimation in this? Because if you add more experimental data, you're constraining these parameters. Is that something that you're planning to add?
Yeah, so Aidan's looking at that. that's definitely on our to-do list. It's quite a big problem. Making a start on it. We'll tell you more about that tomorrow. I think. Or we can chat about it during the breaks and things like that. Okay, let's head upstairs.